Hello, in this video I'm going to talk about sarcomeres. So sarcomeres are the basic functional unit of skeletal muscle tissue that end to end, we put them together end to end to make a myofibril. And then many myofibrils, bundled together inside of a muscle fiber, many muscle fibers bundled together as a fascicle, and many fascicles bundled together as a whole muscle. Okay, so sarcomeres, those are the basic functional unit of skeletal muscle tissue. It's the smallest level of muscle tissue where we have contraction. If we break a sarcomere down into its parts, we no longer have anything capable of contraction. So that's why we call it the basic functional unit. It's made up of thin filaments and thick filaments. The thin filaments are composed of actin, tropomyosin, and troponin proteins. And the thick filaments are made up of several strands of myosin, all bundled together, and then those strands of myosin have little myosin heads that project off from the filaments. So in the picture on the left, you see the purplish part on the left. Those are the myosin strands, and we see the little heads sticking off. And so that whole big bundle, that whole big ropey purple part, that is the thick filament. And then on the right is our thin filament, where we have actin, that's the green part. So it's like two actin threads that are kind of wound together like this. And then we have tropomyosin, that's that kind of orange string that is on top of it. And troponin are like little latches that are holding the tropomyosin down onto the actin. Okay, so the actin protein contains myosin-binding sites that are highly attractive to the myosin heads. So on the actin, there are all these little sites that the myosin heads are very attracted to. So whenever the binding sites are exposed, the myosin heads grab on and form a bond there. So if we don't want there to be contraction, we have to keep those binding sites covered so that we don't have cross bridges forming. When the myosin binding sites on the actin are exposed, the myosin heads bind to the sites to create cross bridges. So at rest, because we want to keep those binding sites covered so that we don't have cross bridges forming, tropomyosin, that yellow or that orange kind of thread there, covers the myosin binding sites so the myosin heads cannot make contact. Troponin, that's the little latch, those little yellow balls in the picture there, so that acts as a latch to hold the tropomyosin in place. Okay, so the troponin, they're just little latches holding the tropomyosin down, and the tropomyosin held in place is covering the myosin binding sites on the actin. So the sliding filament mechanism refers to how the sarcomere actually functions to cause a contraction. So it's the process of muscle contraction caused by the shortening of sarcomeres. And we'll talk about action potentials and neuromuscular junction and all of that in another video. But for now, just how the sarcomere actually functions. When an action potential arrives at the muscle fiber and the calcium ions flood through the cell, the myosin binding sites are exposed. When they're exposed, the myosin heads bind to form cross bridges, and then the cross bridges pull, causing the sarcomere to shorten. Okay, so there's overlap between the thin and the thick filaments, and when there's a signal to contract, the thick and the thin filaments form cross bridges together where the myosin heads are reaching out to the myosin binding sites on the actin, and then they swivel. So they swivel and pull and swivel and pull and swivel and pull and cause the sarcomere to shorten, which is the muscle contraction. Okay, that is all I have for you in this lecture. I'm gonna stop recording here. Thanks for watching.